Okay, so yeah, so let's start from our first topic. I think we have already covered this BS sequencing in the class. So in this lab, I'll basically go over all the things you would need to you would need to finish the homework. The software that we are focused on, there are multiple softwares out there. So there we pick a specific one. It's called Bismarck, and it requires some specific environment, which we also created on in the shared folder, so that you can directly start from there. We think we lock the version, we lock the file writing permission on the shared folder in the shared folder. But please try not to make any change so that the other students can also use the same file. Okay. So the main schedule is basically like this. For the following 2 weeks, I'll be taking charge of the labs. As always, the Tuesday ones will be interactive lab tutorials, and the Thursdays will be the Q&A sessions. So I try to separate the materials into 2 parts so that we can focus on each of them separately. If you have any questions, working on the homework, which by the way, I recommend you to start earlier, please just as always, feel free to use the Slack channel and trying to post it there, and with your screenshot, with your scripts and everything, so that we can take a look at before the lab, before the Q&A session, and also other students from the class can also help, and they can also some of the questions might be you never know, some of the questions may be shared between different students, and you definitely usually not alone. So if you post it there, it will be helpful for yourself and also helpful for the other students. Okay. So the first topic we have here is the BS sequencing. Just a very brief recap of the technology. Is basically using chemical a chemical tool that is you can protect this methylation. You can use this methylation group to protect the C from your genome, and you do this bisulfite procedure, let's say, and then you do all this transcription and duplication, and in the end, when you do this sequencing, you basically change the original genome. You change all the CG pairs into TA pairs, but only those that are protected by the methylation will not be changed, so that will remain the same. So by comparing the outcome, the result genome to the original one, you will be able to see where are the methylation events happening. Okay, so on the right side is a more detailed diagram about if you start from different condition like methylated versus unmethylated one, and then you use the same molecule, is using the same reagent to process. So in the end, if you have this methylation, if you can see my mouse here, if you have this methylation, and that will not be changed, after you do this bisulfite conversion. But if you do not have this methylation group protecting, that will be changed into a TA pair. Okay. So that's kind of the idea behind it. But in practice, now that we know this technology, that could work theoretically, but in practice, what are we really going to do? What would be the difference, from doing, for example, genome alignment and mapping? What would be the difference doing this from what we did before? So we can pause here for a second and think about what would be the additional things we need to do. Apparently, here, since we are going to change the genome, we are essentially also changing the reference, the background behind it. So whenever you do this conversion, you will not be able to realign your sequence, your reads back to the standard genome, because imagine in the standard genome, you wouldn't have such a fragment. You wouldn't have this sequence. So the first thing that if you want to use this BS sequencing, and also for your homework, the first thing you need to do is to make a conversion of your background genome, of your reference genome. So starting from there, Then you would do the regular mapping, regular alignment as before. We can use whatever tools that is available. So here we are going to use this Bismarck software, which is a kind of a bundled up a function where you can do multiple processing steps in one line of command. Okay, so after this alignment, you need to do duplicates removal, extract methylation, and everything. And also this Bismarck also helps you to generate kind of the output report, the log file, which we request you to append into your submission of the homework, so that you don't need to work on that on yourself by yourself. Okay. So the way I want to do this lab is to to lead you guys step by step. Let's imagine we are working on this homework right now. Let's imagine we have a BS sequencing file now sitting on our flash drive. What should we do with it? So what would be the first step? What would be the second? And then I really believe my philosophy is really is learning by practicing. So hopefully through the process you'd also have a better understanding of the theory and of the theoretical algorithm behind it. And also please feel free to interrupt anytime if there's anything too technical or is anything not that clear. Yeah, so first thing we need, as we need before, we need some alignment tool. So we use this bow tie indexing file. I think we used them before. So we are going to use that again. So since we are using this Canon, well, apparently you can use your local machine, but we recommend you to use the Canon server. Then you would have to go through their module manager system. So you would need to load this bow tie, load this package called HSAT and this SIME2, which is a kind of a basic toolbox, say, for genome alignment. For this software of interest, this Bismarck, we leave it to you guys so that you can download and install it by yourself, because this one is not actually a package that need to be pre-installed into your environment. It's more like a folder of commands that you can directly use directly. So as we put in the homework, we ask you to go to this Bismarck document to see how to use them and also how to install them directly from scratch. Just one thing to note here is that this Bismarck software is written in Perl. So try to call it cleverly, because usually the functions we call from the bash, for example, Python here, they are all pre-compiled into a binary file, so they can call this Python as a command directly. But imagine let's say if we go to this Bismarck, so you would have a list of commands that you would need most of them. For example, this one, after you finish doing all this Bismarck thing, you want to generate a report, you want to generate a summary, and you want to call this function. But here you will not be able to say Bismarck, because that's not a command. So in order to compile this thing, you can say there are two ways, basically. There are multiple ways. So the two ways I would recommend is one, to use Perl here. It's just like what we do with R or with Python. We put the command first, we put the language first, and put the script first. Okay, and then that would give you the command and all the help you would need. If you look into this this file, it's actually a script file. So it is actually specified here that to use Perl to call this script. Okay, another way to do it is to specify the full absolute directory, the absolute location of the file. And by doing this, you will also be able to call this function, to call this command directly, because the bash would help you to do this compilation. I see some conversation in the chat. Let me see. Yeah, by default, if we do not specify, try to install everything on your server, in your Canon home folder, or in your Canon wherever you want to put your working directory to be. You can, as I said, you can definitely do everything locally, but we don't recommend that because that would be computationally very heavy, and also it takes a lot of disk space. And also, we already set up this environment on the server, so that we're trying to make a uniform environment so that we can all start from the same page. Otherwise, you might have different versions, and etc. Okay. Yeah. So that's how you call the Bismarck commands. So with all this, I think you'll be able to follow the document that is written here. You know, they have different multiple different tutorials, like step by step. So you just follow their steps. For this genome preparation one, so if you go back to our homework here, we also put in step 1, step 2 things so that you can best stick onto the kind of the plan we expect you to follow. So the first thing you need to do is to download and trying to set up your working environment. And then the second is to prepare for the genome. So here, the genome preparation is the conversion just we just talked about a few minutes ago. Is we put it here as a graduate student only, but for undergrad student, we also highly recommend you to trying to test it out, to check it out, to see, what is really going on, what is this Bismarck is really working on. So, just to give you guys a hint, this function, this command, the entire software is pretty well developed and pretty well maintained. So you can directly use a single command and that give you the preparation. Okay. For undergrads, or if you do not want to worry about this, we also provide a version here. That will be a converted genome version, and you can continue to work on the following questions, just based on this converted one. But again, I would highly recommend you guys to check it out. Okay. Yeah. So actually, by the way, let me show you what is the folder looks like right now, so that you can have a better understanding too. It's taking longer than it usually does. Yeah, so this is what the folder looks like right now. So, instead of having all this markdown file and HTML, we also, in addition, we also have this 3 different data folders and 4 different environments. So try not to worry about the other environments and only focus on this conda environment 37. Because later on, when we do this Python code, especially for the part 2, we would need a lot of dependencies from Python. So you will need this environment. You can activate this conda environment as what we did before. Okay. The thing that is more you care more about is the data folder. So for different questions, part 1, part 2, part 3, we will have different input data. And for example, we just talk about this Bismarck question. So you would see here, for example, this expression location, which is a profile, a background profile of the general gene expression levels for this, I think it's prostate cancer cell line. Yeah, here. So when you see something like, copy the file, you should be able to directly find the files, including all these byte files, all the background gene expression. And here, this HG38 is the converted version of your genome for this test. And if you want to do this conversion, by your own, we would need you to download this the raw genome from the homework one, instead of using the one we provided in P1 data. Okay. Yeah, so after you finish running all this Bismarck, commands, including, preparing for the genome, running the main function, and then just, deduplicate the Bismarck and everything, and, extract the methylation. You just do that. You just follow the, tutorial step by step. And then in the end, you will be able to, yeah, you just do this step by step. I try to put a kind of template of commands here so that you can directly refer to. You'll be able to call this Bismarck report function, and this report will give you something like this. This is, if you see something like this, you succeed. Okay, so this is something we would like to see from your submission of the homework. And then directly from this table, you will be able to directly read, for this specific experiment, what are the percentage of CPGs, what are the percentage of the methylated CPGs, for example, and do we see any clear pattern between CPG and CG, so that you can directly answer the question. Okay. Yeah, so that's all we have for part 1.1. Any questions so far? Okay. If not, let me just pause this recording so that it can be separated into sections.